Hello everyone, hope you're doing well. Today we'll be running through one of the more basic protocols in any tissue cell culture lab, that is feeding cells, right? Really important to make sure that they keep living, keep growing, that they're happy. So you can see at the start here that I'm basically just going to ethanol wipe my hands, spray it with 70% ethanol, rub it in a bit. The purpose of that is because underneath this hood, which is this, space I'm working in, everything's sterile. So there's air blowing in and out that kind of keeps bacteria out, and my hands have bacteria on them, right? Even with gloves. And so to make sure that I use that ethanol spray to basically kill any germs that might be entering the space. Now, this is a simple protocol, but there are a couple small steps, and there's always small things that you need to be focused on, and so we're just going to run through that. In the beginning here, you can see that I'm basically just moving some flasks around, empty flasks. These have nothing in them. These are going to be used for a future experiment. I just kind of prepare them early on by making sure that they're out of the way so that we can keep all of our focus on a clean, open, empty workspace for our cell feeding, right? We don't want to crowd up the space. That just increases the risk for contamination. So I'm grabbing a passer pipette from the outside. This is a 10 milliliter pipette. I'm going to be adding 10 milliliters of media to each of these flasks. That's how much they should have, right? So I've already added it. I'm going to prepare it. You see that I'm making sure to keep that tip away from the wall, away from the floor of the hood. That's really important because if it touches anything, that could introduce contamination. So here I'm attaching a pasture pipette. That's that thin glass needle like pipette. And it's attached to a vacuum tubing, that kind of translucent tubing that attaches basically, that's connected to a waste uh, beaker at the very bottom of the table or of the hood. So I'm basically putting it into the flasks, right? Kind of just touching the liquid with it. It'll kind of suck it out by itself. Don't need to do anything. And yeah, and you can see that I'm being very careful, for example, with making sure that my caps are not face down on the hood, making sure that my pasture pipette tip is not touching anything, it's not touching the outside of the flask. That's all to make sure that it's not getting into contact with any solid surface upon which, bacteria could live, right? Okay, so I'm topping this up to 10 milliliters of media. That's that pink liquid you see there. Was important as well in this step that I didn't show previously, but this media is best stored in the fridge at around, like, 2 to 4 degrees Celsius. That's really cold, right? That's really cold for these human cells, which we're using it in. Human cells work best at 37 degrees Celsius. That is the temperature of our body, right? And so we want to make sure that we are putting these, taking these media bottles out of the fridge, putting them into a hot water bath at 37 degrees Celsius, letting them warm up for, at least 30 minutes before we do any of these steps that I'm showing you in this video, right? I mean, basically just plop it in there, wait 30 minutes, come back and do this procedure. The reason why is because if you introduce these cells to something that's super cold, like 2 degrees Celsius, they're going to be basically temperature shocked, and they're not going to grow as well in the future, right? So just want to make sure that we are keeping them nice and happy, only giving them warm stuff. So just to recap here, I have 10 milliliters of media in each of those two flasks on the bottom. This only goes up to 5 milliliters, and so I'm going to need to take another pipette. Now, I didn't have to. I could just use the exact same pipette, but I'm showing you these steps here because it's good practice to just practice, being very efficient with taking pipettes, in and out of the hood, kind of getting them out of that wrapper, pipetting media in and out. You notice I'm putting it in the biohazard safety bin because it is a biohazard, right? It could have touched potentially some parts of, the cells and stuff like that. And so you want to make sure they're disposing of it properly. You can see here that I'm throwing away the empty media bottle. I'm really tightly capping this other media bottle. Remember, this is all sterile. Should not be exposed to the outside air. Shouldn't have any bacteria inside. Kind of tightening the caps on those flasks just enough so that air it can exchange CO2, but that bacteria can't get in, right? Putting that into the incubator is going to allow it to be at 37 degrees Celsius, their perfect temperature, and also at 5% CO2 in the atmosphere, which is what they work best at. Putting that media bottle in the fridge, and we're done.